Hi students, welcome to Pharm D Guru. Today we will discuss an important topic, inhibition of biliary excretion. If you look at the name itself, biliary excretion means the process of drug excretion through bile. And in this topic, we will see how this process gets inhibited. Okay? Let's first understand how normal biliary excretion happens, and then we will see how it gets inhibited step by step. Normal process. Generally, when you take a drug, it undergoes several phases like absorption, distribution, metabolism, and excretion, right? But at the end of the metabolism, most drugs undergo biotransformation where they are converted into conjugated or non conjugated forms to make them safe for elimination. This whole process occurs inside the liver, right? After biotransformation, the drug molecule moves from the liver to the bile, and from bile it travels to the intestine, and from intestine the drug molecules finally leave the body through feces. Okay, this transfer of drug molecule from liver cells into the bile is what we call biliary excretion. Now let's see the role of transporter proteins in this process. At the surface of the liver cells, there are special proteins known as transporters. These act like delivery trucks that carry drug molecules from the liver into the bile. The major transporters involved are P-glycoprotein, MRP2, BSEP. Okay? When these transporters function properly, drugs are excreted efficiently. But when some other drugs inhibit these transporters, the process gets disrupted, leading to inhibition of biliary excretion. So, what happens next? When the transporters are blocked, the drugs fail to move into the bile. Instead, they remain inside the liver cells and eventually re enter the bloodstream. This increases the plasma drug concentration, which is AUC, to toxic levels, leading to drug toxicity. Now let's see how does transporter inhibition occurs. Okay, the inhibition happens mainly two ways: competitive inhibition, non-competitive inhibition. Let's understand both with examples. Let's see competitive inhibition by taking digoxin and quinidine as examples. Okay, digoxin, a heart medicine, normally uses the transporter P-glycoprotein to exit the body through bile and urine. Now imagine another drug, quinidine, is administered along with it. Quinidine also binds to P-glycoprotein, and both drugs compete for the same binding site. Usually, digoxin has higher affinity for P-glycoprotein, but when quinidine is present, it competes for the same site and blocks P-glycoprotein directly. So as a result, digoxin gets trapped inside the liver, re-enters the bloodstream, and accumulate to toxic levels. This can cause nausea, blurred vision, and even dangerous heart rhythm problems. This is what we call competitive inhibition because there is a competition between digoxin and quinidine, right? Now let's see non-competitive inhibition by taking verapamil and cyclosporine as examples. Verapamil and cyclosporine also affects P-glycoprotein, but in a different way. Cyclosporine does not compete for the active site of P-glycoprotein, but instead it inhibits the energy system that powers this transporter, a process known as non-competitive inhibition. So let me explain this. For P-glycoprotein to push drug molecules from liver to bile, it needs energy, which it gets from an enzyme called ATPase. This enzyme generates ATP, the fuel that drives the transport process. Okay, cyclosporine interferes with that ATPase enzyme. When ATP production drops, P-glycoprotein loses its energy and the transporter cannot move drugs efficiently. So, even though the active site is empty, P-glycoprotein simply cannot walk, meaning the drug stays inside the liver cells and eventually return to the bloodstream, causing toxicity again. Here there is no competition between verapamil and cyclosporine for the active site. Here inhibition occurs through non-competition. So that's why we call this as a non-competitive inhibition. The transporter is inhibited not by blockage but because its energy source is turned off. Now to summarize this topic, in competitive inhibition, two drugs compete for the same transporter site. Example, digoxin and quinidine, right? In non-competitive inhibition, a drug disrupts the transporter's function instead of competing for the site. Example, cyclosporine interfering with the ATPase in P-glycoprotein. Both conditions slow down biliary excretion, leading to drug buildup, higher plasma concentration, and finally toxicity. And that, students, concludes today's topic, inhibition of biliary excretion. Thank you for watching. Yours, Pharm D Guru, signing off.